Hi everyone and welcome to another class discussing virus reproduction. We've always wondered, how do viruses reproduce when they're not even living organisms, which means they do not fulfill any of the 7 life characteristics in Mrs. Gren: movement, respiration, sensitivity, growth, reproduction, excretion, and nutrition. The only thing they can do, by the way, is reproduction. But how does this happen in viruses? Well, they are considered from the obligate intracellular parasites. And this terminology you should know, by the way. Obligate intracellular parasites, which means they can only reproduce within a host cell because they do not have the cellular machinery nor the materials to synthesize more viruses. They can enter a host cell in a variety of different ways. They can affect 3 types of organisms: bacteria, animal cells, which includes human cells too, and plants. For the bacteriophages, they inject their genetic material into the bacteria. However, the animal viruses enter the cell via endocytosis by fusing their viral envelope. They fuse their viral envelope, of which we called the lipid envelope, made from proteins and lipid, with the host cell surface membrane. So the animal viruses enter by endocytosis, and this occurs by the fusion of the viral lipid envelope with the host cell surface membrane. The third thing is the plant viruses, and they use a vector such as an insect as the aphid. You guys know it? An insect such as the aphid to breach the cell wall in order to penetrate the cellulose strong cell wall. We have to do something to that. Once inside the host cell, one of the following pathways can occur. It's either the lysogenic pathway or the lytic pathway. Lys means to break or to cut. Genic means to the genes, the genome. However, lytic is just cutting. And we're gonna discuss what the terminology means in here. Lysogenic pathway and lytic pathway. For the lysogenic, it has a phase where the virus remains dormant, which means it doesn't do anything to your body. There is no symptoms attached, no symptoms accompanied to that virus. They do not immediately cause the disease. However, they cause a disease. It's just not at the moment. Once they infect a host cell, the viral DNA is known as a provirus inserted into the host DNA. But a viral gene coding for a repressor protein prevents the viral DNA from being transcribed. That's how there's a delay, and this is called latency, which you can call it's sleeping, it's dormant, it's not doing anything valid that affects us in a negative or even a positive way. And the time during which it occurs is known as a period of lysogeny, which we derived this name from, lysogenic pathway. Viruses in a lysogenic state may become activated and enter the lytic pathway. So, what we've just concluded that there's nothing that happens during the lysogenic pathway. Everything can happen only during the lytic pathway, and this occurs when the virus, which is in the lysogenic, moves into the lytic pathway. Activation may occur as a result of host cell damage or low nutrient levels inside a cell. It becomes activated ever since the host cell damages, which, as an example, you can take the bacteria. If the cell surface membrane of the bacteria ruptures or lyses, this is a clue that the host cell damage caused it to be moving into the lytic cycle. So the whole thing is that there is a repressor protein that prevents the viral DNA from being transcribed and translated. And why do I even transcribe it or translate it? This is certainly because I need to synthesize the body or the components of a virus. Components. As we've seen before, clearly in this diagram of a bacteriophage, which is a type of virus that is from the DNA viruses. As you can see, it has components such as the capsid, nucleic acid, tail tube and sheath, long tail fibers, spikes, base plate, and collar. All these have to be synthesized individually. Have you heard it? Individually. So I have to synthesize, transcribe them first, then translate them into these components of a bacteriophage. Then we combine them all together to form this virus. That's why ever since it has been produced as repressor proteins, ever since the repressor proteins were produced, this prevents the viral DNA of the host cell from forming these components. But they can be reduced as in concentration. The repressor protein concentration may drop or lower in number as a result of host cell damage. Now moving to the lytic pathway, then we're going to summarize both. Okay, as you can see in here, the lytic pathway, where the viral genetic material is transcribed and translated to produce new viral components. Finally, it can be transcribed and translated to produce our new viral components, of which we discussed in here. These components are assembled. We connect them just like Lego blocks. We connect them to form the whole virus into mature viruses that accumulates inside the host cell. Finally, the host cell bursts, and this makes sense because the whole pathway is called the lytic pathway, where the lysis of the cell surface membrane occurs. And this releases a large number of viruses, starting from hundreds to thousands and millions of viruses, each of which can infect a new host cell. So each one of these can inject all over again their genetic material into a whole new bacteria and cause this cycle to continue all over again, causing more cell deaths, cell ruptures. Okay, the host cell bursts, which releases a large number of viruses, can infect a new host cell, and this is called as lysis. This is what typically results in a disease. When you ever experience coughing, sneezing, or feeling ill and sick from a specific type of virus, this is because we're in a lytic pathway of a specific virus. Now let's sum up what we've said in these two diagrams and compare between lysogenic and lytic cycles. Firstly, lysogenic cycle occurs, of which there is more or less nothing happening to the host cell. There is totally nothing happening. The virus is dormant and it doesn't cause any effects. This is peaceful. Nothing happens. As you can see here in the steps, firstly, the first step is common in both lysogenic and lytic cycle. The phage attaches to the cell surface of the bacterium. The bacteriophage attaches to the surface, and here too, the bacteriophage attachment and inject the viral genome into the cell. Here too, it's gonna inject its viral genome into the cell. The phage DNA enters the bacterial cell, and in the lysogenic cycle, it becomes integrated into bacterial DNA. Integrated here means incorporated, which means it got stuck with it as part of it. It's incorporated. Okay? Here, this part is just like it's part of the DNA. Then, the integrated prophage replicates when bacterial DNA replicates. And here we stop until some certain conditions, such as the cell death, occurs so that the concentration of their repressor proteins decreases and they can finally be activated to cause a disease. That is when it becomes violent and it turns into the lytic cycle. So again, a summary of the lysogenic cycle. The phage attaches to the cell surface of the bacterium and injects its DNA to enter into the bacterial cell. It becomes integrated or incorporated into the bacterial DNA. Then replication occurs when the DNA replicates, followed by the host cell damage when the virus is in the state to be activated. The amount of repressor proteins decreases and the virus enters the lytic pathway. This is not the end. This is just the start of another cycle called the lytic cycle. And the lytic cycle can occur on its own, not followed by, or like not the precursor of lysogenic cycle, whatever you call it. Here, at first, in step number 1, attachment and injection of viral genome into the cell. It attaches, then degradation of the host genome. The host genome is going to be damaged. You see how bad is this lytic cycle to us or to a certain bacteria? It injects its genome inside, the genome enters, and then right after the genome enters, it's gonna damage the host cell DNA. Then, after it's damaged, the cell components of the bacteria, such as the ribosomes, are going to work for synthesis of the proteins in the components of this virus. It's working, but not for itself. It's blindly working. It doesn't know. It's not doing it for itself. It's doing it for the virus. Okay? Synthesizing the individual parts of the components of the bacteriophage, as you can see, here is the capsid and the viral DNA, the spikes and everything. Then it's gonna assemble it and release it. The release actually occurs due to lysis of the cell surface membrane in here. The cell surface membrane of this particular bacteria is going to rupture and lyse. That's why we call it the lytic cycle. So this results in death of the host cell. Of course it dies because the cell surface membrane is ruptured and new viruses are released, which are going to progress and move on their lives by infecting more bacteriophages. Here is a final summary of what we've said in a single diagram. And here, as you can see, we have both the lysogenic cycle and the lytic cycle. First of all, we are in this first step, of which the bacteriophage is going to inject its DNA into the host cell. Then it either becomes incorporated in the cell surface membrane it enters by endocytosis, as we've just said, and it's going to be incorporated within the bacterial or the host cell's DNA. In here, it becomes incorporated. Then there's either a chemical trigger that kills or not kills, actually breaks down or digests this DNA. It digests the DNA, and this is where we are in the lytic cycle. It digests the DNA of this host cell and then is going to use the components of the host cell, such as the ribosomes and other components, to form the building units of this bacteriophage all over again. So now they are totally formed, and the new virus particles are going to burst out of the host cell. And this is the lytic cycle. It could have just undergone another pathway, of which, ever since it is incorporated in this cell in here, it's not activated by a chemical trigger anymore. It is going to be replicated. After being incorporated, it's replicated into more cells. Here, as you can see, the host cell replicates and there are 2 cells. The 2 cells are gonna remain inside them the viral DNA, but it's dormant. It doesn't function until there is a specific thing that occurs, such as bursting of the cell surface membrane, or something like the host cell is damaged, the repressor proteins, which prevent it from transcribing and translating the components of this virus, are going to be reduced in concentration. The repressor proteins are less until there are none, and it moves again like that in here to the chemical trigger and enters the lytic cycle all over again. I hope you guys have understood this very well, as it's an important topic in her syllabus. Thank you for watching.